Hello, in this video we will discuss about the physiology of mitochondria. The physiology of mitochondria is more diverse, but we will understand in this video just the basic concept. So here is a cell contain also the Golgi bodies. This is the nucleus, the plasma membrane, and the inner side is the cytoplasm, cell membrane. And this is the Golgi bodies, the mitochondria. Mitochondria contain a double membrane organelle like a chloroplast. And in this way, here is the inner matrix, outer membrane, inner membrane of the mitochondria. So this is the double membrane organelle. Let's remember and let's begin to understand the couple electron proton transfer through NADH oxidation and quinine. And basically we are just going to understand the electron transport chain with explanation. So remember, first is the complex 1. This complex 1 is the this is the inner mitochondrial matrix, and this is the inner mitochondrial membrane. So this is the outer mitochondrial matrix and the outer mitochondrial membrane I am not drawing here. So just remember that. And in this way here is L-shape protein, which that is the enzymatic protein, which that is known as basically used for the couple electron proton transfer through NADH quinine oxidation. So the quinine oxidation and quinine reduction is involved for this because the quinine is a protein, is mobile protein. And this is the L-shape NADH dehydrogenase enzyme, which that is used for the transmission of the proton to the quinine to reduce into the quinine H2 or ubiquinine. So in this way here is the NADH will be oxidized to the NAD plus, and this FMN protein will receive this proton to transmit the iron sulfur protein with the electron. Basically it receives the electron, and the iron sulfur protein will give the donation of the proton to the quinine H2, the quinine ubiquinine to the H2, and again this reaction can be cycle occur. So remember, this 4 proton will be pumped out. How it is possible? Let's begin to understand. This is the FMN bridges. This basically NADH2 electron donor with the iron sulfur 1 electron acceptor. This is the L-shaped complex 1, which that is basically overall reaction we will learn about. This is the NADH and quinine, and 5 proton is required to form NADH plus QH2, and 4 proton will pump out. And in this way the flavin mononucleotide oxidized to basically 2 electron and 2 proton will donate to the FMN to form the reduced FMNH2. The flavin mononucleotide after reduction to form a FMNH2, which that actually donate the proton to the Q pool, the quinine pool. So in this way, this is the cytochrome BC1. This is the complex 3 involved first, and next we will discuss about the complex 2, because this is reason. So this is here is the QH2 will donate to the cytochrome B the electron and proton to pump out, while here is the quinine H2 will also produce from the succinate dehydrogenase enzyme will donate to the cytochrome B2 to produce 4 proton pump out. So remember, the 2 proton is involved to produce from the succinate dehydrogenase, but it will not pump out, but it will donate to the cytochrome B, to iron sulfur protein, to cytochrome C, the electron acceptor, the cytochrome C, while the proton will be pumped out. While here is the QH2 is involved for this reaction to the FADH2 will oxidize to the FAD plus, and the iron sulfur protein donate to the QH2, and the cytochrome C is donate to the electron to the complex 4. This is the complex 4, which that cytochrome C is the mobile protein with the copper A protein and the cytochrome A3 and QB, which that is accept the electron, while the proton will pump out here from this mechanism. And in this way the 4 proton will pump out, while the electron will be lose their energy, and the half molecule of oxygen and proton will reduce into the water. This is the condensation reaction. And in this is the cytochrome oxidase enzyme, because it is used for the oxidation of the cytochrome. So the oxidation of the cytochrome will release the electron, and in this way the free electron will be released in the form of water. So let's begin to understand the ATP synthesis. That is the complex fifth is known as the ATP synthase enzyme. This contains two domains, F1 and F0. F1 is the inner matrix domain, while the F0 is the membrane embedded domain. So in this way, basically F1 is contain polymeric, mean the alpha beta subunit, and between the alpha and beta subunit, basically the epsilon and the gamma subunit are present, and in this way the delta subunit is also attached here from this, and it is used for the stability of this, for the rotation, and on the other hand the ATP will be synthesis from this F1 domain. While this is the G-arginine 210 is involved for the alpha subunit will lead to proton pump out because it creates a positive charge will lead to move inside, because the inside is the proton less, so it is allowed to proton move inside, and this is the B2 domain subunit, and in this way this is the rotation of the gamma rotation due to the proton passes makes a gamma spin within alpha and beta head. So remember, this is the subunit inside is the F0 containing 10 subunit. The basically is the decameric subunit. So this is the binding change mechanism, ATP synthesis. In this way the ATP will be synthesis, we will discuss now. So remember, the outer mitochondrial matrix basically contain less pH, means the high proton will rush from the alpha subunit of the ATP synthase enzyme to pump inside, because the movement due to the gradient constriction again toward the gradient where the pH will be increased. So this is the open, new ATP is released, and ADP and inorganic bind is the O side, so the open side when after releasing bind again new ADP, and the loose side is the bound ADP and inorganic phosphate cannot be released, and in this way is you can see this the ADP and the inorganic phosphate is the loose side, and in this way remember inside is the gamma subunit is rotating for creating the when the proton will move out. So the tight condensation of ADP and inorganic phosphate is favored to form ATP. The ATP forms is very tightly. So remember, this is here is the T site, which that is tightly bound, and basically it transmits toward the O site, and this O site before the moving to the O site the ADP release and the ATP release and ADP will add. So the pump out the proton will prefer the gamma subunit rotation lead to synthesis of protein. So synthesis of ATP, not protein, sorry. So in this way here you can see this. Let's begin to understand also the physiology of uncoupling protein UCP thermogenesis. So how the thermogenesis will occur, how to produce heat from the UCP protein. So basically this is the same mechanism. The oxidation reaction will lead to proton pump out, while the basically proton pump out the ATP synthase enzyme used to produce synthesis of ATP. And remember, while this is the adipose tissue, in the adipose tissue as a adipocyte will break down to during the cold weather, and lipolysis and peroxide anion HFA fatty acid containing, which that will be move inside into the inner mitochondrial matrix. After this the beta oxidation will be occur, and the Krebs cycle will lead to produce more NADH as well as other things. So remember, and in this way here I am drawing, this is a proton, this is a proton. Basically proton will also move into the UCP for when move inside, because the outer side is the pH will be less more acidic, while the inside is the more basic. So the move toward the gradient constriction again toward the when where the constriction will be less, so the osmosis like semi permeability are present, and in this will produce heat when proton will move in. While on the other hand, the fatty acid is also used when moving inside. The beta oxidation can be possible, will lead to proton production, as well as the other things which that will lead to move out. The fatty acid will produce more heat, and this is the Krebs cycle. The beta oxidation will lead to produce the acetyl CoA, and the acetyl CoA is used for the Krebs cycle to synthesis more NADH, and FADH2 will use for the electron transport chain. So anyhow, I hope you make sense about these things. The couple electron proton transfer through NADH, and basically is the quinine oxyreductase, and the FADH function for the succinate dehydrogenase enzyme. And this is the basically the physiology of mitochondria overview. Thank